Welcome to David Charis Pharmacology. In this video, we are going to be learning about pharmacokinetics, drug metabolism. When we talk about drug metabolism, we are looking at the transformation of drug or the breakdown of drug by the liver or the kidney into its inactive form for easy excretion. Let me just put it here to be easily excreted. That is for drug metabolism. But I'm going to be looking at basically the metabolism of drugs and the factors that affect drug metabolism. But we know that for every drug that gets into the body, the drug might be an active drug that has no active actions. The drug might be converted from an active drug to an inactive drug. It might be converted from a prodrug to an active drug. It might be converted from a toxic substance or a toxic drug to a non-toxic drug, which can be easily excreted as urine. I'm going to be focusing on this active to inactive drug, or active form to its inactive form. So basically, let me just clean this. So basically, when you take a drug and this drug is now in the bloodstream, for example, you take a drug orally, it will encounter the liver. And remember from our definition of metabolizing of a drug by the liver, breakdown of a drug from its active form to inactive form by the liver is referred to as first pass effect. First pass effect. Because this drug, the first passage of this drug is by the liver. That is why it is called first pass effect. And it's undergoing metabolism, also known as first pass metabolism, before it is now some of the drugs are distributed into the bloodstream, while some of the drugs are distributed to the kidney. The kidney further metabolizes the drug into its inactive metabolite and excreted as urine. But yeah, I'm going to be looking at the liver and important metabolizing agents or important metabolizing structure that causes this metabolism or first pass metabolism, first pass effect, any of which you choose to use. Now, in the liver, we have an enzyme called the CYP450 family. The CYP450 family is a group of iron-containing enzyme, Hem. Remember, hemoglobin, right? So it's a group of Hemi-containing enzyme that metabolizes drugs. We have we have discovered like about 60 different families or so, but there are some that are like present and functional in mammals. But this CYP450 family, in full form, cytochrome 450. 450 is the relative molecular mass or so. Cytochrome 450 family. Now, we have different types of cytochrome 450 family enzymes. These enzymes are the ones responsible for metabolizing of drug. They are the ones responsible for first pass effect. We have different units. We have the CYP2E1, we have CYP3A4, we have CYP2D6. CYP3A4 metabolizes most drugs. At least 60 to 70% of drugs is metabolized by CYP3A4. Now, what does 2EI mean? What does CYP3A4, CYP2D6, we have CYP2C19, we have CYP2C9. CYP means cytochrome, that means it belongs to the cytochrome family. The 2, which is the second number, that means it belongs to the second family or second group. The third one, it belongs to the D subgroup. That is the subgroup family. while the sixth or last one is the isozyme. We don't really need further details about this, but just know that these are the, and we have others, CYP2C19, we have other CYP450 family. So basically, the metabolizing of drug undergoes two main process, or um two two main conversion so we have the first one being the first conversion, which is the phase one transformation. The first one being the phase one transformation, and the second one being the phase two transformation. Now, metabolism sometimes can be interchangeably used with biotransformation, the conversion of a drug from one form to another, as in this case, from the active form to its inactive metabolite or inactive form. So the phase one biotransformation of a drug is the metabolizing of the drug by CYP450 enzymes, CYP450 enzymes. Now, this CYP450 family, these enzymes are present in the smooth endoplasmic reticulum, SER. Remember our cell physiology, smooth endoplasmic reticulum of the hepatocyte of the liver cells, hepatocytes. They are present in the smooth endoplasmic reticulum of the hepatocyte. And the phase one biotransformation processes that occurs there are oxidation, reduction, and hydrolysis. Phase one transformation seeks to convert active form of drug, which are polar, to inactive form. Active form of drug which are non-polar to inactive form which are polar. Phase one biotransformation of drug. So we have the CYP450, undergoes or ensure that these drugs are undergoing the process of oxidation, reduction, or hydrolysis, either by adding a polar group to it, polar group like sulfate group, hydrogen, hydroxyl group, and other polar molecule, so as to make the drug more polar so it can be easily excreted. That is for phase one biotransformation. And in phase one biotransformation, there are several factors that can affect this biotransformation process, affecting the overall metabolism of the drug. We have what we call genetic polymorphism, and we have what we call CYP450 inducers and inhibitors. So the first one I said, still under phase one, I said there are factors that affect phase one metabolism or affect metabolism as a whole. We have genetic polymorphism, we have CYP450 inducers, and we have CYP450 inhibitors. Now, what is genetic polymorphism? It's like from the word polymorph. Morph means structure, right? Poly means different or many. Genetic polymorphism is the change in structure of the CYP450 enzyme. So imagine I have the CYP450 enzyme now, and I have something attached to it. I'll call this a CYP450 polymorph. Now, this polymorph can influence drug in the sense that it can, when this CYP is metabolizing a particular drug, this polymorph can chew through the drug. It can, fasten its metabolism. So individuals with genetic polymorphism of a particular drugs are what we call, fast metabolizers. They can be fast metabolizers of a particular drug, and they can be slow metabolizer of a particular drug based on their type of genetic polymorph. that is being attached to CYP450. Some polymorphs are, they increase the metabolizing activity by assisting the CYP450 enzyme in metabolizing drugs. Some polymorph inhibit the enzyme as a whole. So drugs are poorly metabolized. So this makes individuals with genetic polymorphism to various drugs either fast metabolizer or rapid metabolizer of drugs and slow metabolism of drugs. Let's talk about the CYP450 inducers and inhibitors. Now, certain drugs or certain substance, when they are taken, they actually induce the CYP450 enzymes and they also inhibit the CYP450 enzymes. CYP450 inducers, they are drugs that increase the metabolizing activity of the CYP450 enzymes, increase CYP450 metabolism or metabolizing activity. An example of drug inducers are carbamazepine, carbamazepine, phenobarbital or phenobarbital, phenobarbital. We have rifampin. Um, we have amphetamines and so on and so forth. Why drugs that decrease the CYP450 metabolism are the CYP450 inhibitors. An example of a drug are omeprazole, ketoconazole, fluconazole, erythromycin, gentamicin, basically majorly the antibiotics, the aminoglycosides, and the rest and the likes. And of course, don't forget an important um, fruits, because there might be food to food interaction, food to drug interaction, that some food like grapefruits can inhibit CYP450 enzymes. And grapefruit basically inhibits the CYP3A4, a CYP450 enzyme that metabolizes almost, most of the drugs that we take. So grapefruit is a major inhibitor. That is all for the phase one biotransformation. For phase two biotransformation, we are now looking at the conversion of this drug that have undergo phase one, that is now, so let's say this is a drug, the drug have undergo phase one transformation and the drug have become polar. Phase two transformation or phase two biotransformation now take this polar drug or now take this metabolite from phase one biotransformation, converting it into a more polar form that can be easily excreted. Remember, when the drug is polar, it it cannot absorb, it cannot get back into the blood circulation because that drug is polar. So when we have a more polar form of drug, it can be easily excreted. So I have phase one transformation or phase one metabolite of drugs undergoing um, conversion by phase two into a more polar form. In phase two biotransformation, an important process that occurs there is what we call conjugation. Conjugation is the, from the word conjugate, the joining of a polar molecule. So conjugation is the joining of a polar molecule to a phase one drug or if yeah, a phase one metabolite that is already polar but not so polar to be easily excreted to form a more polar excretable molecule. And it does this by a series of transferase enzymes. That is the definition for conjugation in phase two biotransformation of drug. Now, what are these conjugate enzymes? I hope you get all this process. Conjugate enzymes like um, let's let's take the first one, methyl It undergo phase 2 transformation, undergo the process of methylation, acetylation, um, sulfation, glutathione addition, gluta glutathione addition, glucuronidation addition by their enzyme methyltransferase. We have the glutathione, you know, transferase enzyme, glucuronidation transferase enzyme. I think the full enzyme is UDP. Yes, uridine diphosphate glutathione transferases. You can check your textbook for more. But basically, this process is methylation adds a methyl group to a particular drug, making it more polar to be easily excreted by the kidney. Acetylation add acetyl group, a polar molecule to a particular drug. The same thing for sulfation, add a sulfate or a sulfur group. The same thing for glutathione, it add thiol or SH to be easily excreted. Glucuronidation also add a glucuronide, which is a very polar molecule to a particular drug, so it can be easily excreted. That is all for phase 1 and phase 2 biotransformation or metabolism of drugs.